Okay, this is component 2, paper 3, upper 6 mark. So, the diagram below represents part of DNA used to introduce a gene into a bacterial plasmid. So we're talking about splicing into a plasmid. And we've got base sequence there, region X, and I don't know whether they're going to ask me to do stuff with the bases, but we'll see. Name the type of enzyme that was used to remove the DNA. So that's a restriction endonuclease, which is its proper name, and occasionally on mark schemes you'll see a restriction enzyme, and restriction endonuclease is its proper name. Why not use it? Not that difficult. Okay, name the type of enzyme that was used to join this DNA into the plasmid. So here we're talking splicing, and that's DNA ligase. Super duper, 2 AO1 marks. Name the regions labelled X. So these are overhanging single bases after you've made a cut. Cut your DNA out. Okay, this is the gene that we're putting in our plasmid. These are the overhanging ends. Those are the entertainingly named sticky ends. Named by a biologist, that, definitely. And what's the role of those regions? So, the unpaired bases are complementary to those on the plasmid. So they join the gene into the plasmid. They didn't give you a lot of room to write that, did they? Okay. The following is a list of mRNA codons and the amino acids for which they code. Great. The upper strand of the inserted DNA, shown in the diagram, was used in the synthesis of a polypeptide. Name the first 2 amino acids. So 3 bases code for 1 amino acid. So this is DNA, complementary base pairs, G, G, C, C, A, C. Okay, so GGC, glycine. Let's try using the synthesis. Name the first 2. Which way we're going? Let's go that way. GGC, and CAC. Glycine, glycine, GGC, glycine, glycine, and CAC, CAC, nothing else, no, CAC, and histidine. Name the organic base represented by G. Again, spelling important. Guanine. Give an example. So again, this is kind of all AO1 this, of where a human gene is introduced into a bacterial plasmid for medical use. So I'm talking anything else apart from medicine. So to produce insulin, or of course the example in your booklet, human growth hormone. if you know something about perhaps cystic fibrosis, you could put, to produce alpha-trypsinogen. FSH, presumably they make this way. And give two advantages of this technology. So this is more AO2, isn't it? A bit of AO3 perhaps. So so what can we say? So we've taken a human gene, we've put it into a bacteria. The bacteria is then churning out a human hormone. So, and it is churning it out. So let's do that. So we get insulin. I'm going to use my insulin example. Is produced rapidly in large quantities, so better than using a pancreas. The insulin is human insulin. So there's no rejection of the protein. Not seeing it as being foreign. And I suppose let's put an ethical one in as well. No need to use animal products. What's the problem with that? Which may be carrying infection. I'm not going to feel too sorry for the animals because they're probably being killed for meat anyway. Okay.